Welcome, everybody. My name is Harmony Peraza, and I'm the Study Participant Manager for the Dog Aging Project. I'll be your host today as Amber enjoys some vacation time away. So welcome, everyone. It's wonderful to have you here. I absolutely love seeing those pictures of all of our pack members in the slideshow. Everyone was looking great, some very cute holiday sweaters and lots of great pairs of dogs and dogs enjoying the outdoors and all that. It's wonderful to see you all there, all your pups. If you're a member of the pack, please be sure to join us at the dog park. That's our private community. It's a forum just for pack members. That's where you can share pictures of your special pack member and chat with members of our research team. So always remember that it's there and head on over. You can also ask questions for our guest speakers today and enter our monthly giveaway at the dog park. There's still time, so head over to the dog park forum now. We have an exciting presentation for you today. We're going to be chatting with members of the Cornell Veterinary Biobank. We wanted to give you all a look behind the scenes at the Dog Aging Project and the Biobank and what an important role that it plays. We can only imagine looking forward what new technology and new methods will be available in the future. We have questions such as, will there be artificial intelligence to help us analyze DNA? Will a discovery change the way that we analyze blood serum? We're curious about all of these things. Keeping valuable samples safeguarded at the biobank lets scientists in the future attempt to answer questions we haven't yet even thought of. So we're very thankful to be working with the Dog Aging Project Cornell Biobank. Before we get to our speakers today, I would like to give you just a little bit of background. As you know, the Dog Aging Project is a research program where participants primarily contribute observations about their companion canines. We collect your observations using a series of very important surveys. We collect the observations from tens of thousands of pack members, and we appreciate your efforts in those very much. So our researchers are then able to make scientific discoveries with just the power of paying attention to what you offer us in those surveys, so thank you. However, we're also able to add more precise information by collecting biological samples from some of our participating canine companions. Handling specimens is much more labor intensive for us than collecting survey answers. So we're not able to do this with just with everyone. This means that we have scientifically represented. This means that we have to use a scientifically representative cross section of our pack members. We call this smaller group the precision cohort. Members of the precision cohort are invited to participate by our research team. And the invites are still going out. So don't worry, there's still a chance that some of you may be invited to the precision cohort. And if you agree to be a part of this smaller group, you'll be asked to have your veterinarian, collect some biological samples from a kit that we send you. We mail that to you in the mail, and then those in that cohort would take that on to their veterinarian and have their veterinary team collect those samples. And we will work with you to get that timing right on getting these samples collected via an appointment that you make. We typically recommend you align that with your annual exam for your dog. Then your veterinarian will use the contents of our sample collection kit to collect samples such as urine, feces, blood, and hair samples. And then we'll send that to our lab, or your veterinarian will send that to our lab using a FedEx label. The whole process is very routine, safe, and quick. The samples will then be processed by our lab, and the researchers will get to work. However, there's sometimes material left over after processing those samples, and that's where the biobank comes in. Any biological material submitted by our PAC members that remains after the study will be kept in a long-term storage at the biobank. So now that you know the journey our samples take in getting to the biobank, please let me introduce our speakers and let them talk about the important work that they do. The Dog Aging Project Biobank is operated by Cornell University's College of Veterinary Medicine. Dr. Reba Herndon and Dr. Laura Moutan join us today from New York.
Good afternoon.
Good afternoon.
Welcome. Thank you. Welcome. Thank you for joining us from so far away. I guess we're really all across the country, so thank you for being here. So just quick introductions here. Dr. Moutan is a laboratory coordinator for the Cornell Veterinary Biobank. She has a background in cryobiology and reproductive physiology with 6 years of experience in biobanking. Dr. Moutan oversees the laboratory operations at the biobank for the nucleic acid processing and banking, and that is a part of the quality assurance team. And it helps the biobank maintain its ISO accreditation, which is very important. She assists the Dog Aging Project by managing the Dog Aging Project Biobank at Cornell for storage and distribution of the biological samples, which are collected throughout the project, and by providing guidance on quality management. And then also joining us is Dr. Herndon, and she is the Quality Assurance Manager for the Dog Aging Project Biobank at Cornell University, the teaching hospital there. And she is an experienced quality and education professional with expertise in process improvement in both industry and academia. Dr. Herndon has over 15 years of experience in quality management systems and over 20 years experience with process control, process discipline, and training adults in the workplace. She has led a high impact research and development teams at companies like Boeing and Corning Inc. Dr. Herndon is responsible for supporting the quality efforts at the Dog Aging Project Biobank through scheduled internal audits and continuous review of the biobank's processes and procedures. You two ladies have extremely important jobs. We appreciate your passion for quality. We're so happy to have you here and learn about the biobank.
Well, thank you very much for having us here. We're really excited to be here.
We are.
And actually, today we have a guest, a very special guest that we invited to come join with us today to talk to you a little bit more about the DAB Biobank. But unfortunately, she's a little bit shy and she really doesn't like to speak in public. So Harmony, we were hoping that you would be able to help us out with our guest today.
I certainly can. I'd be happy to lend a hand. Thanks for calling on me.
So here she is.
It's Allie Q, and she's an aliquot. Hey, Allie. So the first question that Miss Allie Q has for us is, what is biobanking and what does it have to do with the Dog Aging Project samples?
Well, that's a great question. And biobanking is really the preservation of biological material for future use. And biobanks, the way that they work is that they're repositories that specialize in the acquisition, storage, and distribution of biological samples. They can also perform the collection of the samples themselves or process the samples like doing DNA extraction or adding preservatives, or test the samples to get more metrics of quality for them. But that really does depend on the biobank themselves. Not all of them do all of those functions. So biobanks can store a wide variety of samples, including microorganisms, plants, animal samples, human samples, or even environmental samples. And the stored samples can be used for scientific research, clinical procedures, or conservation efforts. You might know already a few different types of biobanks, including the Red Cross Blood Bank or an umbilical cord bank that stores cord blood samples from newborns to be used for stem cell research, or the Svalbard Global Seed Vault located in Norway, which is used as a backup for the world's crop diversity, or even the Cancer Moonshot Biobank that was recently started by the National Cancer Institute, which stores biospecimens from human cancer patients to help accelerate cancer research. So Reba and I are part of the Cornell Veterinary Biobank, which has been a core resource at the Cornell University College for Veterinary Medicine since 2006. And we are located in Ithaca, New York, in the beautiful Finger Lakes. What we do, our mission really, is to serve the research community by helping accelerate research as an accredited resource for standardized biospecimen collection and archiving. And the way that we do that, it really is with the goal of improving both animal and human health by promoting translational research, which is also called bench to bedside research, and that will ultimately result in more precision medicine advancements with more effective, better targeted, and less toxic therapies. So we do this by collecting or receiving DNA and tissue samples from many different types of animals with different disease profiles. We mostly work with dogs, cats, and horses, but we also have samples from a wide variety of different animals, both wild and exotics, and domestic species as well. And after we've received the samples, we process them, we test them, store them, and then we're able to distribute them out to researchers. So one thing that I do want to let everyone know here is that even though we are a veterinary biobank, the research that is done in animals can also benefit human medicine. The traditional strategy is that research is done in the lab, and then a treatment can be developed and tested in humans, and then the treatment is applied or modified for the dog, for example, in which case animal medicine is always behind the times compared to human medicine. But there is a newer, better strategy that has currently become more widespread, which is to do the research at each level and to use the knowledge that we've gained from each of those steps to benefit the others. So for example, if we're studying lymphoma in the dog, it not only helps us treat the disease in the dog, but also it can be used to develop treatments in humans and helps us understand the disease as a whole better. So everything that we do here for DAP not only benefits dogs and all our dogs that we know and love, but also they are able to help us with human diseases in the long term. So now that you know a little bit more about biobanking, I want to talk to you about the DAP Biobank. And so the Dog Aging Project has been teaming up with the Cornell Veterinary Biobank to create the DAP Biobank, which is really just a branch of the Cornell Veterinary Biobank that is dedicated to DAP specifically. And so after we received the samples from the DAP participants, they send us the samples that are not immediately used for the DAP projects. And the samples that we receive include whole blood samples, serum and plasma, and also PBMCs, which are peripheral blood mononuclear cells. So they're essentially white blood cells. We're able to then store the samples and then distribute them out to researchers to help them accelerate their research. And the reason that why DAP does this, instead of just using the samples for their own plans that they have, is to really be able to accelerate the research all around the world, not just what DAP in particular has for plans for. And one reason why they're using the Cornell Veterinary Biobank to help them with these banking and distribution efforts is that they want to make sure that the researchers have access to the highest quality of samples possible.
All right, we have another question. My dogs are chiming in the background. Why is quality so important?
Quality is important to us, no matter how big or how small your dog is. Quality means handling your samples consistently, doing it right the first time. Quality management involves quality control and quality assurance. Quality control, or QC, is focused on fulfilling quality requirements, checking and to make sure our freezers are at the right temperature. And then quality assurance provides confidence that the quality requirements are fulfilled. Quality assurance, or QA, is using the right procedure and equipment, and that means making sure our thermometer is calibrated and reading the right temperatures. Quality control and quality assurance together make up our quality management system. The Dog Aging Project Biobank is an ISO, International Organization for Standardization, accredited biobank. And the Cornell Veterinary Biobank was the first in the world to be accredited to this standard. We also use standard operating procedures, SOPs, which are detailed documentation on how to do what we do so well. Helps with consistency in our process. Enough of all that, Allie Q. Let's break it down and talk about quality for the rest of us. I call it the grocery shopping analogy of quality. When you shop for groceries, you have your list. And believe it or not, you use your quality standards such as no broken eggs or outdated produce. No wilted lettuce, ugh. And your milk and cheese should be at the correct cold temperatures. Nobody wants warm milk or broken eggs in the box or rotting, slimy vegetables. After all, we have standards. There's a concept I'm going to introduce you to. We're going to use a concept called PDCA. Plan, do, check, act. We're going to plan, we make that shopping list. We're going to do, we're going to go to the store. We're going to check, make sure that everything we buy is fresh, the right temperature, and our shopping list acts as a checklist to make sure we don't miss anything or forget anything. And then we act, of course. We buy those fresh vegetables, that cold milk, 12 eggs, not 11 eggs. Using something like plan, do, check, act is a great way to mitigate risk or mistake proofing for what we're doing.
Okay, Allie Q has another question for us, and it is, what happens to the Dog Aging Project samples once they arrive at Cornell?
That's a great question. So first, the samples arrive at the Texas A&M lab for the Dog Aging Project in Texas. And once they are done with using the samples with what they need for the project, they are shipped to us in Ithaca. And of course, it's not a straight line because, well, you know, shipping companies never do that. So the package really does a trip all around the country before it gets to us. But it does get to us in a timely manner. And also, depending on the types of samples that are in the package, we ship them either with ice packs or on dry ice to make sure that they are maintained at the correct temperature so that by the time they arrive to us, they are still nice and cold, just like they're supposed to be. Now, once they are arriving in the lab, we accession them into our Biobank Information Management System, or BIMS. And that is really just a database that helps us keep track of all the biobanking information that is pertinent to those samples. We're able to get that information directly from DAP, and we are able to supplement it with all the information about the samples as we process them. So for all the samples that we receive, we get 1 tube of blood, and we use that for DNA extraction. And that is going to be helpful for us to be able to do sequencing on those, for all those dogs that are participating in the precision cohort. After we extract the DNA, it is being stored directly at -80, which is a very, very cold temperature in our new freezer, which is called the SAM HT. We also receive 1 other tube of whole blood, and this one gets aliquoted out, which means to be portioned out into smaller little parts. And those samples get stored also directly at -80 degrees Celsius for future use. We receive the serum and the plasma samples that are already prepared by the team at Texas A&M, and all we need to do for those is store them. And they also get stored at -80 degrees Celsius, just like the blood and the DNA. And the last type of sample that we receive are the PBMCs. And these ones actually get stored in liquid nitrogen and the vapor phase of liquid nitrogen, which is much colder. It goes all the way down to -190 degrees Celsius. And that is basically what happens to all our lovely samples here.
All right, another question, very important one. How do you ensure quality during this process?
That's a great question, Allie. Quality is ensured through careful handling of your samples, and we also do traceability of the samples. Quality goes in before it gets stored at every step that you saw earlier in this presentation. Cross-checking the name of the animal with what sample is being processed. Handling each sample with care in a consistent, documented manner. All of our equipment is calibrated and certified. Our thermometers are calibrated, our pipettes are calibrated, even our centrifuges are calibrated so they spin at the right speed using a calibrated tachometer. Thanks, Allie, for helping me talk about traceability. Traceability is the ability to track and trace the history, application, and location of a sample at the biobank. We use specific labels and containers for your samples using our documented process, SOPs, standard operating procedures. Samples are appropriately tagged so that we can identify them throughout their time at the biobank. We use printed labels, barcodes, and a 2-dimensional identifier. These samples are linked to documented information with detail on permissions or restrictions associated with the sample. Next, we use our BIMS, Biobank Information Management System. We talked about that earlier as well. That helps us edit and ask questions of the information associated with that sample. It includes, but is not limited to, collection, transportation, preservation, and storing. This system has a built-in alert for any deviations from our biobank procedures. We establish a link between the sample and the data in the form of a unique biobank ID. That helps us maintain it for unmistakable traceability. This allows us to identify the location of any sample and its data at any time. We always are looking for ways to improve our quality processes. Quality is a continuous improvement process. You make corrections along the way, you learn from experience with each step, and identify other actions and add to your quality improvement strategy. Lara, Allie Q, and I, along with the Dog Aging Project and Cornell Veterinary Biobank team, thank you for your time. And a special thank you to Deborah for helping us with this presentation. We would be happy to answer any questions from the audience. Thank you.
Thank you so much, Dr. Herndon and Dr. Mutam. We do actually have some questions from the chat, and let's just start at the top here. One great question is, why do samples need to be stored at different temperatures?
That is a great question. So generally speaking, the colder the storage temperature, the better it is for the samples, but we get at a point where you have to balance the costs involved in it. So for example, storage sample in liquid nitrogen temperatures, so very low temperatures down to minus 196 or 190 degrees Celsius, it's a lot more expensive than storing them at minus 80 degrees in an ultra low freezer, which is still more expensive than storing them in a regular freezer, which is minus 20 degrees Celsius. So for what we have here, for the DNA samples, the whole blood samples, serum and plasma, storage on minus 80 degrees Celsius is sufficient to ensure the quality of the samples for many years to come to be used for research. The PBMCs, so the peripheral blood mononuclear cells, because they're actually preserved in the way that keeps them alive, it's much better to be able to store them at very low temperatures to ensure that they do stay fully frozen and that once we warm them up, they can stay alive. And the reason we do that is because then researchers can use those live cells to study different aspects of aging or disease in the dogs. It kind of creates almost like a snapshot in time from when the samples were taken from the dog.
Okay, excellent. Thank you, Dr. Mutam. If we have time for another one, let's see here. Can you talk about the type of technology you use to store the samples?
Sure. So I already mentioned a little bit about how we store our samples at minus 80 degrees. And we actually do them now using a new technology, which is automated storage. So what you can actually see here, what we have here, it's called a Hamilton SAM HD automated storage system. And it's really great new technology that allows us to automatically store samples with very little human manipulation in the process. There's multiple aspects of that that are very beneficial. One is that it simply helps keep the samples colder by not having an actual freezer door being opened in and out. So we're not letting cold air out and hot air into the freezer when we do that, because it's got special gates happening throughout the system to block out the air. Another aspect of that is if you noticed at home that you get frost buildup in your freezer, that happens when you have humid air that comes into your freezer, and then the water in the air gets condensed into ice, and that's where you get frost buildup. Because of this type of system, because it really controls the environment, it only lets dry air going in, which means that we don't get any frost buildup in the freezer. This makes it really safe for our samples to be stored in them. It also means that whenever we go to take out samples, instead of having to take out an entire box out of the freezer and pick out the samples we want and exposing all those other samples still to the room temperature air, the unit automatically cherry picks exactly the samples that we need one by one and delivers them to us. So we're really protecting all the samples that we have stored by having them in this system. It also just makes it a lot safer also for our staff, because then we don't have to be handling samples that are that cold, because minus 80 degrees Celsius is very cold. It is dangerous to touch samples at that temperature. So it really limits the contact that we, the staff, have to have with them and keeps everyone safe involved. Also, because it uses barcode systems to track everything, it means that we're able to create lists and have them being picked automatically based on barcodes. We're reducing the human error aspect of reading a number wrong and picking the wrong tube out or anything like that. We generally have processes, we call them buddy systems, of having people double checking when we do this, but having it on top of it, intelligent automated systems that help us do that really cuts in on the work. It cuts down on errors and really makes it simpler for us to be able to handle these samples. So that's one way that we're getting new technology brought into biobanking and brought to the DAP, to the dog aging project, to really help make sure that we're providing the highest quality of samples to the researchers, that we're really doing it in the best and most efficient manner that really helps everyone around.
The Hamilton freezer sounds like an incredible piece of advanced technology. Although my German Shepherd, oh boy, would complain that you couldn't leave the door open and grab some ice cubes. So he just wanted me to mention that. Okay, wonderful. So Dr. Herndon, maybe this one might be for you. In the chat, someone is asking about the quality aspects you've presented. Are those typical for research projects?
Actually, they're not. The DAP Biobank and the Cornell Veterinary Biobank are pretty unique in their ability to embrace quality. It made a lot of the things I do a lot easier. They had a well-documented process, and they had the mindset about quality. They really embrace going from unconscious incompetence, doing things like, we're doing it wrong, we don't know, to conscious incompetence, we're doing it wrong, but we're starting to document, to conscious competence. Not only are we doing it right, we've got it documented. And what I love about this group is they have moved to unconscious competence. It's like breathing to them. It's a part of their culture. And that is unusual for many research projects.
Thank you so much for that, Dr. Herndon. We'll try to move along and get to as many questions as we have. And so another one from the chat today, one of our participants is asking if samples lose any quality with the shipment from the original clinic to Texas and then to New York. And how do you confirm the sample quality before it's used at the biobank? Two-part question.
Well,
You want to answer that one, Reba?
they don't lose any quality as long as the temperature that they were originally stored at or refrigerated at is okay. And our dear shippers haven't lost them. And we do have ways to check the integrity of each sample. And I'll have Lara tell you about that.
So one thing that we do, for example, is that when we receive the samples at the biobank, we measure the temperature of the samples on arrival. And so that confirms that they haven't warmed up too much in transit. So that's one way that we verify that they're being kept at the correct temperature the entire time. And then we have quality steps that we've put along the way to check the quality of the samples. A lot of times we just do spot checks because the actual quality test destroys the sample. So we can't test every single one of them, but it gives us a representative idea of how well all the samples are doing in that way. We've also, before the start of the project, have done some small pilot studies to test out how the entire process of going from the vet to DAP at Texas to the biobank, how that entire process affects the samples. And we found that it doesn't significantly affect it. It's true that the samples would be at the highest quality if they came literally straight out of your dog and straight into a freezer, but that's just not feasible for the scale of this project, this massive, amazing project that we're doing. So what we're doing is the best that we can under the circumstances. And more importantly, we record absolutely every step of the process so that if a researcher for their specific needs, they need very stringent quality requirements, we can tell them, we can let them know exactly what happened to that sample, and they can decide if it is good enough for their research or not. But at least they are informed and they're able to make conclusions on their research based on that information.
You've got an incredibly detailed process there. That's amazing. Well, we'll get to the last couple of questions here. And one participant is asking how many samples can be stored?
So in this one unit, the SAM HD, the way we have it configured, it can take up to about 80,000 samples. If we were to switch it to a slightly different setting, which would hold essentially more of the smallest tubes possible, I think it gets up to over 100,000 samples, but you need to really pack them in there in very specific arrangements. So essentially the size of that unit will hold enough samples for the entire project for DAP of what we have projected.
Okay, that's incredible. Finally, we're curious, what is the coolest type of sample or animal that you've banked?
Reba, what's your favorite animal we have banked?
It is one of my favorite animals, the two-toed sloth.
We do have some DNA samples from two-toed sloths. But that is really what we have stored at the Cornell Veterinary Biobank, so the broad biobank rather than the DAP one. If at DAP, I would say every sample is my favorite because all of your dogs are just as equally special to us. That's a cheesy answer.
It's true.
Well, that's certainly a common theme for us because we adore our participants and their companion canines and enjoy talking with them and holding these events monthly. And Laura and Reba, we thank you so much for your time today and this fantastic information that you've shared with us about the Cornell Biobank and specifically the DAP Biobank at Cornell. Just a couple of thoughts here for the group. It's important to remember that we hope what we can discover about the aging process in our companion canines through the health surveys and through the biological samples that will be collected at the Dog Aging Project Biobank can one day be translated to healthy aging in humans. That's really important to remember. And again, the Dog Aging Project Biobank being, it is operated by Cornell University's College of Veterinary Medicine, has the highest standards of and passion for quality. And we appreciate our relationship with you all. So from here, our participants can read and share the article about the biobank, and you would find that website at, find that article on the biobank at our website, dogagingproject.org. We really thank you, our PAC members and broader group, for joining us today. This has been a very enlightening discussion. We've had a great time. If you didn't get your question answered during the live event, please stay tuned. Our team will be answering the questions on the Dog Park Forum throughout the week. And don't forget to enter the giveaway at the Dog Park also. Eric will be announcing winners there very soon. If you missed any part of today's program, the video will remain available to replay on your YouTube channel. And as always, if you or someone you know hasn't yet joined the Dog Aging Project, it's definitely not too late. Recruitment is ongoing, so you just head over to our website, dogagingproject.org, and click the Nominate Your Dog button and join the pack. Again, we appreciate you all for joining today. We thank you so much, and we can't wait to do this again next month with a new and exciting topic. So looking forward to it. Thank you all.